Hello everyone, I'm Steven and you're watching Steven Loves Science. And in today's video we're going to gain a beginning level understanding of the process of transcription, or the process in which genetic information is transcribed from DNA to RNA, which can then later be translated into proteins or what have you. Like I said, this is going to be just going over very beginning level concepts. I mean, transcription is really very complicated. I mean, you have gene regulation, which determines which genes are transcribed. It's all very complicated. I'm not even going to cover enhancer regions, which actually activate what is called the transcription initiation complex. But, I'm going to save a little snippet of that for the end, okay? All right, so let's begin with the animation. Okay. So really what we're doing here in transcription is the goal is to produce mRNA. So we're producing messenger RNA, which then in cells that have nucleuses is going to leave the nucleus and going to travel to the ribosome, where it'll then be translated into functional proteins. Hopefully functional proteins. So as I said, the synthesis of messenger RNA is called transcription. And transcription begins when RNA polymerase recognizes and binds to a specific promoter region along the double-stranded DNA molecule. A particular subunit of the RNA polymerase, called sigma factor, participates in recognition of the promoter region. So it's going to help RNA polymerase find the promoter. So soon after transcription is initiated, the sigma factor dissociates from it dissociates from the RNA polymerase. It says, goodbye RNA polymerase, I helped you find the promoter, you don't need me anymore. So now the RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded mRNA. Now, I just want to go over this briefly. The template strand, a complementary strand is going to be formed off of the template strand. So, as we can see here, which I pulled up in the animation, if we look closely, we can see the familiar base pairing rules apply, the same as DNA to DNA, but in DNA to RNA, there's a small exception to the rule. Now, thymine does not exist in RNA, so thymine is replaced with another base pair, with another base called uracil. So where in DNA adenine would bond to thymine, in RNA adenine bonds to uracil. I'm not exactly sure why that is. It's pretty interesting, but if you know why, leave a comment in the comment section below. Okay, so we can see it's the same base pairing. So cytosine is bonding to guanine. The only difference is instead of thymine, you have uracil binding to adenine. So as we discussed with the DNA replication video, polymerase enzymes, so in this case RNA polymerase, synthesizes in the 3' to 5' direction, with the new nucleotides being added to the 3' end of the growing RNA molecule. So as the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. Now, when RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the mRNA, and that causes the mRNA molecule and the RNA polymerase to dissociate from the DNA and fall off. Now that has to do a lot with something called a Rho protein, RHO, but you can look that up on your own time. So RNA polymerase and the DNA molecule dissociate from the DNA, thus completing transcription. Now, transcription, at this point you have an RNA molecule, but it's not very useful to the ribosome. If the ribosome got this RNA molecule, stuff would be all over the place, and you wouldn't have very good proteins. So then what happens is a process called RNA splicing. And then since an extremely small portion of our DNA actually codes for functional proteins. So during RNA splicing, what is called a spliceosome complex forms, and that cuts out what are called exons, and those exons are non-coding regions of DNA. So those exons are then taken out, so only introns are translated by the ribosome. And we're going to take a brief look at actual transcription in real time. It's an animation from DNALearningCenter.com, but it'll give you a good view. So we have two perspectives of transcription today. So let's take a look at that. Okay, so right now we're looking at the promoter region of the DNA, and following that promoter region is the gene. So first things first, as we discussed earlier, the sigma factor is going to bring over the enzyme RNA polymerase, which we should see coming up shortly. So there we have sigma factor bringing RNA polymerase. And now what I talked about before were these enhancer regions, and we're going to see those in a second, but currently we have forming what's known as the transcription initiation complex, which is all the proteins and enzymes needed to start transcription. But following much upstream of that, it can be hundreds or even, I believe, thousands of base pairs upstream, is what's known as the enhancer region, which is going to carry a protein that initiates the transcription. It's called an activator protein. So there we have the activator proteins that actually has to touch the transcription initiation complex to start transcription. So now here we're going to have that starting now, and that immediately sends RNA polymerase racing off, and it's just creating all the mRNA at this point is being synthesized. Okay, so we can see here the RNA nucleotides being brought in through the RNA polymerase's intake hole, and it's synthesizing mRNA. So just a good recap on what we were just discussing. Alright, okay, so thanks for watching. I hope this helped, and as always, subscribe, rate, and comment. Hello everyone, I'm Steven, and you're watching Steven Loves Science. And in today's video, we're going to gain an intermediate, a beginner level understanding of mother